Greetings all, and welcome to today's presentation. Today we will be talking about the efficacy of using rapid ABO blood group genotyping using high-speed droplet allele-specific PCR, and more specifically, how the technology responds to using crude blood samples for a clinical setting. ABO genotyping is an essential technique in many fields because this technology does not require the use of fresh blood samples. This study offers a new way in which ABO genotyping is possible at much faster speeds and comparable accuracy, all while using non-ideal blood samples. They attempt to do this by using droplet PCR technology. This technology is a previous creation of the authors and has allowed them to get PCR results at much more preferable speeds. To understand this process, we must understand just a few working components. The ABO gene has various single nucleotide polymorphism sites, or SNPs. And at each of these sites, there are two possible sequences. As an example, here at Table 1, we have two variations, G and delta G, of one of the SNPs. This process was called droplet allele-specific PCR, and it allows us to test which or both variations are present. And doing that across a few SNP sites will allow us to reference the ABO genotype. Allow me to explain just a bit further. There are three of these SNP sites that we must consider. At each SNP site, there are two variations that are all tested. Using known expression patterns from these six possible present alleles, we can reference the genotype with very high accuracy.
Hi, my name is Carlos, and I'm going to go into the materials and methods to droplet PCR. So for the materials, droplet PCR, it kind of varies depending on the protocol you're following, but here are just some common materials. PCR primers, which have specific DNA sequences with the target allele. You can also get DNA samples, which is just genetic material with the target alleles. You can also need a PCR master mix, which is a mixture of components such as like DNA polymerase, DNTPS, buffers, and stabilizers. And they just help with the reaction. Fluorescent probes, which are helpful in labeling the specific alleles. In this case, we do use some microfluidic chips, which is a device to help create smaller droplets for the reaction. And then usually for PCR reaction, it's common to have a PCR machine, which is just helpful in amplifying the DNA. So you also need some type of detection equipment for the fluorescence, something to analyze the amplified droplets, and you can tell if the alleles are present or absent, and then some software for the analysis portion, a program that can interpret the fluorescent data and determine genotypes. So for the methods, you kind of start with sample preparation. For our case, we're doing crude samples. So we're doing extractions from blood, saliva, tissues. And when you're getting it from the source like that, you can avoid the whole DNA purification step. So this is what kind of helps speed up the process. Next, you design a primer. So something that has that target and desired genomic region that you're looking for, and you have fluorophores as well attached to that. Then you go ahead and add the PCR master mix, which is what has that DNA polymerase, DNTPS, buffers, and stabilizers. They help mix with the DNA sample, and then they also help with partitioning it out into tiny droplets. And you can kind of see that here with the oil phase. So next, you go into the amplification step. So you take the sample and you put it through some thermal cycling. So the PCR machine or a thermocycler is what you use to help amplify the target DNA region. So for every droplet, it's being amplified. Then you go into the fluorescent detection. So you go into the droplets and see which ones are emitting a signal. If it's emitting a signal, that means it's the target allele is present, or it could not be present, depending on what you're kind of going for. Then you do a data analysis. So with that fluorescent data, you can use a specialized software to either detect if the allele is present or if it's absent. You can even see frequency and the concentration as well.
Hello everyone, I'm Wenwen Liu. Here's the result. Specificity of the droplet-based PCR method for ABO genotyping using genomic DNA extract from fresh PB. Figure 2 are represented amplification plots were shown according to the criteria in Table 2. In SNPs, the droplet-based PCR provide amplification for 261G is in AO, BO, and AB types, but not in the OO type, where only 261 delta G was present. Amplification for 261 delta G was observed in AO, BO, and OO type, but not in AB type, where only 261G was present. Amplification for 526C and A03G were detected in all genotype tests as expected. Amplification for 526G and A03C were detected in BO and AB type, but not in the AOO type. All of the genotypes determined by the droplet-based PCR were concordant with the genotype from the direct sequencing method. All amplification were observed within 8 minutes. Detection limit of droplet-based PCR for 3 SNP loci. By the serial dilution experiment, the droplet-based PCR could detect 261G, 261 delta G, and A03C at allele ratio of 5%, and 526G is 10%. The AA and BB type were not available, so author evaluate the detection limit for 526C and A03G using plasmid DNA with 5% and 1%. A representative experiment with the AO genotype is shown in Figure 3. DNA concentration was shown here. The specific amplification for all 3 SNP loci using crude DNA from buccal cells and dried blood spot samples show the same amplification as DNA extract from PB. The SNP pattern was compatible with the AO genotype. All amplification were observed within 9 minutes. The genotype determined by the droplet-based PCR method were always consistent with those obtained by direct sequencing.
Hi everyone, my name is Madeline and I will be going over the conclusion and significance of this research paper today. To summarize, the method of droplet allele-specific PCR enabled rapid PCR amplification of crude DNA samples that were not purified. Crude DNA from buccal cells and dried blood was pre-treated with proteinase K in a digestion buffer. This method of ABO genotyping was able to yield results in less than 9 minutes and performs as well as the traditional direct sequencing methods that can take over an hour to amplify the DNA tested. The droplet ASPCR method enables a high specificity for amplification of desired templates and is able to avoid false DNA amplification, which can occur in conventional PCR. The method presented was able to provide specific amplification using allele-specific primers for the single nucleotide polymorphism sites at position 261, 526, and 803 in exons 6 and 7 of the ABO gene. These 3 SNPs were enough to identify the A, B, and O alleles which occur frequently in the test population. The significance of this method can be seen in the field of forensics as well as in clinical settings. In forensics, this method allows for crude DNA samples from crime scenes, such as blood, hair, or small tissue samples, to be quickly and accurately genotyped. Unlike conventional PCR methods, the rapid droplet ASPCR method avoids false DNA amplification in the presence of impurities. In clinical settings, this method of ABO genotyping has many beneficial uses. ABO genotypes are used as donor-recipient discrimination markers in transplantations, and rapid ABO genotyping is especially useful in human leukocyte antigen-matched but ABO incompatible transplantations. Rapid droplet ASPCR can be life-saving when used to detect fetal maternal ABO incompatibility. Rapid genotyping allows for early detection, diagnosis, and treatment of hemolytic disease of the fetus and newborn. Rapid ABO genotyping using allele-specific PCR has proven itself as accurate as conventional PCR methods and allows for the use of crude DNA. The speed, sensitivity, and specificity of the droplet ASPCR method for ABO genotyping makes it incredibly desirable in clinical settings. Thank you for listening to our presentation today. For further discussion, how does droplet ASPCR compare to other genetic testing methods used in clinical laboratories? And what are potential implications for wider adoption of this technique in a clinical setting?